Hi, my name is Audrey Tepper, and I may look like a park ranger, but I'm actually the historical architect for the National Malls. At around 2006, the National Park Service started to notice that there was a strange growth on the white marble of the Thomas Jefferson Memorial. Around 2016, we were working with some microbiologists who identified this as a biofilm. Each year, since about 2017, Dr. Burnett and her students have visited the Thomas Jefferson Memorial. They have collected samples, very sensitively taken, of the biofilm. They have brought them back to their the laboratories at the high school and have sequenced the DNA.
The biofilm project has been really, really cool in that it has produced a huge amount of data. And what I've been trying to do is see, are there better ways, other ways of analyzing this data than what we've done in the past and what scientists have been doing in the past?
The National Park Service was great. So they did clean the whole memorial in 2019 with laser treatment, but they left specific dirty spots along the radius of the dome for us to collect samples from. We run it through the sequencer to find its bacterial and fungal compositions.
The machine is going to go ahead and then shotgun all of the DNA that you've collected and basically look for one specific area of, well, not really one area, several specific areas of one specific gene of interest. Instead of looking at this in terms of taxonomy, I basically used machine learning to go ahead and take all of those, 300 base pair long DNA sequence reads and then reduce them into actually two-dimensional points that you can visualize on a scatter plot.
We found this specific fungal species called Ascomycota is promoted by oil or petroleum pollution specifically, and so we hypothesized that because DC is like a really crowded metropolitan city that has a lot of cars, trains, trucks that all use petroleum, this increased pollution could have increased Ascomycota and increased the biofilm.
I think honestly the personal genome machine was the most impressive and also intricate machine that we've been able to use. It looks at the pH changes of the DNA samples when it attaches the nucleotide bases, and that's how it figures out what the actual sequences of DNA are. So I think that's really fascinating.
We had a hypothesis because previous years studies had looked at biofilm on the memorial and they had found species that were native in Japan, and we thought that these species could possibly have come from cherry trees because the cherry trees were originally a gift from Japan. So we sampled cherry trees at the memorial in the Tidal Basin, and we also grew our own cherry trees at TJ as kind of a comparison point.
There's this specific bacteria called Crocroxidiopsis. That this species is found on almost all of our samples that were taken from a Tidal Basin tree. They are also found on almost all of the untreated samples that the other group took from the Jefferson Memorial. And this has potential to show that there maybe is stuff moving from the trees in the Tidal Basin to the memorial, and it creates a baseline for future projects to look into this in more depth.
Doing science in the lab is really exciting and fun, but it just makes it more real when you see that it has real applications in national monuments that thousands of tourists go to visit every single day.
Right, that's something that I think that is unique about TJ, is that I get this opportunity to go out and to be aided by organizations like the National Park Service to actually complete a project that has an impact on the real world.
I mean, data is cool. That's, yeah. it wouldn't be possible without access. You have no idea. We have literally gigabytes of data that have been produced by that sequencing machine. It was a valuable experience. I don't think I could have gotten it anywhere but here.
The National Park Service is really lucky to have the Thomas Jefferson High School of Science and Technology on their team to study the Thomas Jefferson Memorial. I tip my ranger hat to the students at the high school who have just been terrific, and to their fearless leader, Dr. Mary Susan Burnett, who has really been a phenomenal partner in all of this.